Hello, my name is Claire Bamber, and this is my project on the age of genetic exploration and discovery. Key individuals and groups like Avery, MacLeod, and McCarthy, James Watson and Francis Crick, and the Genome Theory Project group led to the age and exploration of genetics. The rise in the study of genetics is mainly occurred around the second half of the 1900s and continues on today. The discovery and exploration of genetics is so important because it impacts the medical, agricultural, and historical fields that are improving and changing our life and humanity daily. The story starts in a kind little monastery garden in the St. Thomas's Abbey of the Czech Republic, where a monk named Gregor Mendel grows his pea plants. This is where Mendel founds the beginning of the science of heredity and genetics by discovering the pattern of heredity in his pea plants. Though he is not an American, he's still one of the founders of genetics, the same science field that will be further explored and become influential about a century later. As we move on a century later, we can see that the advances of the microscope then move the progress onto the 20th century. Around the mid-20th century, they already knew the presence of cells, DNA, protein, other organelles, and genetic material. But where is genetic material? Scientists were conflicted on the whereabouts of heredity. Most thought it was in protein, but a small group of scientists, including Avery, MacLeod, and McCarthy, discovered that the actual location of genetic material is within DNA. This discovery rose another question. What is DNA made of? Many were curious about this question. It only took a group of non-geneticists named James Watson and Francis Crick, with of course the support and information from Rosalind Franklin, Erwin Chargaff, and many other scientists to piece together the puzzle. With the X-rays they collected from Rosalind Franklin and the knowledge of nitrogenous bases from Erwin Chargaff, Watson and Crick were able to put together the correct structure. They ended up with a structure of an anti-parallel double helix made of 2 strands of nucleotides, all connected together. In the end, it looked like a spiraling staircase. Now that scientists know the location and the structure of genetic material, they can now move forward to figure out how DNA transfers its blueprints to program life. Well, the answer to this question is in the process of central dogma. This process starts with DNA transcribing itself, then turning into RNA. Then it leaves the nucleus and passes through these organelles called ribosomes, which translates the information from RNA to make proteins. Then these proteins go off to do millions of different processes and functions. The sequence of DNA and RNA are extremely crucial because even one letter change can affect the makeup of an organism. While scientists in the 1970s were studying the reaction of a bacterial cell under viral infection, they realized that the way the bacterium was protecting itself was through the use of an enzyme. This enzyme had the ability to cut through viral DNA at a certain location. These are called restriction enzymes. Scientists were fascinated by this discovery and decided to try to use these restriction enzymes to be able to reproduce large amounts of specific fragments of DNA. After scientists located the type of DNA, they cut it out with restriction enzymes and put it in the DNA of a bacterium. After that bacterium multiplied, Then scientists can splice it out and have a large amount to study the organism's genetic sequence, expression, and its evolution over time. This technology even allowed for the creation of insulin medication for diabetes. As this technique developed, scientists were able to use these restriction enzymes to copy whole sequences of an organism's genome. We currently have more than 100 organisms' genomes copied. Then, in 1990, an international group of scientists created the Human Genome Project. Their goal was to map out the entirety of the human genome, and then they officially completed the rest of the genome in 2003. As new discoveries are found, new ideas also are found, which comes to genetic engineering. But what is genetic engineering? Well, based on the Essentials of Genetics 6th edition book, genetic engineering refers to the alteration of the genetic makeup of living organisms using recombinant DNA technologies. Now you can find genetically engineered or genetically modified organisms, also known as GMOs, almost anywhere you go. More than a dozen of the crops that are genetically modified are verified to eat and buy, with many more in the process of being verified. Again, in the book of the Essentials of Genetics, it quoted that 60% of the processed food in the United States contains ingredients from genetically modified crop plants. This topic on GMOs can be controversial, and I can see why that would be. But I do believe that GMOs are helpful and revolutionary, especially with the world's current population, illnesses, conditions, allergies, and deficiencies. For example, scientists are currently trying to fight deficiencies like vitamin A by producing crops that has an increased nutritional value based on their staple food. An example would be golden rice. Cloning has also become popular in this field, especially with the first clone of Dolly the sheep in 1996, starting this idea of a desirable offspring. Scientists have made herds of animals that have human proteins in their milk. This can result in a commercial use of drugs and medication. It even goes as far as the U.S. Supreme Court saying that you can patent these animals. With more than 10 million people in the U.S. suffering from a genetic disorder and 3% risk of a child being born with genetic disorder, genetic engineering can be applied for gene testing and gene therapy to help those who are suffering from a genetic disorder. Parents now have the ability of having insight before they bear a child to see if they could carry anything like a genetic disorder onto their child. The evolution of genetics over the past century has been absolutely amazing. It has accomplished and unlocked numerous branches of fields, creating new frontiers of discoveries and ideas to explore. Scientists have solved the biological makeup of ourselves and almost every organism on the planet. And along with that, they also established a way to alter this makeup of life, which is absolutely phenomenal. The field of genetics is daunting for sure, but we can only hope that scientists will use this knowledge to help society with more of their amazing discoveries.